Hello, everybody. Welcome to this webinar for the Association of Physicians. It's a pleasure to have some very good speakers here. All of whom are from Oxford, working on different aspects of inflammation and different sort of clinical to kind of the lab approaches, but all with really good impact. And hopefully everybody will find the way they all work together really attractive and informative. So I'll hand over to Chris Buckley, who's key to all of this. And then at the end, when they've all given their talks, there'll be an opportunity for some Q&A. And so if you've got some questions, just put them in the Q&A box, or if you can't find that, put them in the chat, and we'll try and make sure they're all answered for you. So over to you, Chris.
Thanks, Paul. What has a rheumatologist, a pediatric gastroenterologist, and a bioinformatician got in common? Well, they're all interested in inflammation. We're going to tell you a little bit today about the problems with the way in which, not the definition of inflammation, which is as old as the Greeks, cardinal features of hot, swollen, painful, loss of function, and then the understanding of the cellular basis of it. But how do we work across different diseases which share inflammation as a common theme? And so we're going to present kind of 3 15-minute snippets. Holm is a professor of pediatric gastroenterology and director of the Center for Human Genetics in Oxford, and he's going to tell us a little bit about how genes interact with environments to kind of give you the outcomes that are important. And genes often come out early in the story because, of course, in children, that's the part that they first manifest themselves. But some in adults, sometimes people with rare diseases can present later. Callie is going to tell you a little bit about how understanding perturbation in studies, what we're calling cell atlases that really look at therapeutics. So therapeutic cell atlases can play a role, and all you ever wanted to know about that. And then I'm going to end with a little bit about so what. I mean, how does this knowledge actually change the way we practice and look at medicine? So it's a great pleasure to introduce my two other colleagues. As I said, Holm is a pediatric gastroenterologist, and he'll kick off. We won't take any questions after each talk, but Paul will coordinate this at the end, and then Callie, and then me. So Holm, take it away.
So thank you so much, Chris. And I'm just putting on the slides. Do you see slides?
Yeah, they're good actually, and they're full screen, which is fantastic. Thank you.
Perfect. So what I like to discuss is this interaction between genes and an environment in the pathogenesis of immune-mediated diseases. And I want to focus on one group of diseases that are inflammation of the gastrointestinal tract. And we have IBD, inflammatory bowel disease, two main disease groups, Crohn's disease and ulcerative colitis. And many people sort of have a bit of a concept to say, well, if inflammatory bowel disease happens to children, in particular to infants, then it must be genes. And if this happens to adult people, in particular elderly people, then it must be more environmental factors. And in fact, sort of this sort of is partially true, but in many cases it is both. It is genes and environment and how they do interact. What epidemiological studies have told us that many different factors can lead and contribute to inflammatory bowel disease. These can be stress factors, smoking does contribute, appendectomy, antibiotics, our food, so many different factors. And although epidemiology sort of isn't causality, it leads to some of the really intriguing questions. So, for instance, smoking is a risk factor for Crohn's disease, but it's actually a protective factor in ulcerative colitis, and we do not really know why. Similarly, sort of an appendectomy can be a protective factor, for instance, in ulcerative colitis. And again, we sort of observe this, but we don't fully understand the mechanisms. What seems to be a reoccurring feature is that our food, all factors that affect our food, strongly sort of influence the susceptibility to inflammatory bowel disease. And this can start from birth and the gestational age, delivery mode that does affect the microbiome, but also our diet, infections, use of antibiotic have an impact on the microbiome. And then different bacteria produce metabolites, and one of these metabolites is butyrate, which is produced, for instance, by certain bacteria, for instance, Clostridia or Faecalibacteria. And so of these short-chain fatty acids have a major impact on multiple cells of the gastrointestinal tract, for instance, the epithelium that is required for maintaining barrier function, macrophages or dendritic cells that take up, for instance, bacteria, can present it to cells of the adaptive immune system, and then sort of cells of the adaptive immune system like B cells, plasma cells that produce the immunoglobulins, or T cells that are conductors of the immune system. So in this way, sort of it's an explanation how variable factors, including antibiotics or the food, basically sort of lead to an modulation of the microbiome, and then sort of how this leads then to metabolic rewiring of the microbiome, and these metabolites then can have an impact on the immune system, and that feeds back in a cycle towards the microbiome. In addition to these environmental factors, there are hundreds of genetic loci that have been linked to inflammatory bowel disease, and we currently know over 300 loci in our genome that are associated with disease susceptibility. That includes sort of variants that are protein coding, but also variants that are intronic or even intergenic in this setting. Genetics has been proven extremely complicated to understand, in particular since the majority of variants are indeed intronic or intergenic. And to give you one exemplar of how these genes, or many of these genes might act, this is an exemplar published last year by the group of James Lee. And what this group, Stanke et al., have described is that the gene locus that is linked in gene regulation that affects an enhancer setting, and by affecting an enhancer that sort of has an impact on multiple different genes, one gene locus can have a significant impact on macrophages and dendritic cells, on their response to microbiota, on their production of inflammatory cytokines. And this is one locus. It affects multiple different genes. It affects the response to the micro environment, and that is a reoccurring scheme. In addition, what genetics tells us that some of the genetic loci that have been identified are indeed regulated in the epigenetic setting as well. So they highlight how sort of genes and environment intersect. And one of these locus is a gene where sort of not much information is actually available. SPNO2, less than 30 publications on these genes overall. And here we have sort of an indication about genetics, about epigenetics, and we recently identified as these genes again has an impact on how macrophages handle, for instance, bacteria. As Chris has alluded, in the extreme young age in pediatrics, we see not only this polygenic disease group where many different genes contribute to disease, but a group of gene defects that have a very strong impact, and we call them monogenic. We call them monogenic because in one or two variants in a gene, typically protein coding, has a major impact on this barrier function, on T cell immune regulation, or on macrophages, and this can lead to a high penetrance of disease. And as an exemplar, if there is a gene defect in the immunoregulatory cytokine IL-10, then this leads to inflammation, typically in infants with 2 to 6 months of age, so even in babies. But knowledge here has expanded as well, and we know now over 100 monogenic conditions that can lead to IBD as a consequence of a primary immunodeficiency, and this can lead to a number of extraintestinal manifestations as well. Often these patients with these gene defects are very difficult to treat with conventional medications. Some of those require, for instance, hematopoietic stem cell transplantation. Now, one of the key questions that the field has is to understand why some of the common variants, for instance, can lead to inflammation. And an exemplar here is a gene that has been identified 2 decades ago. It's called ATG16L1. And one variant there, in particular, this T300A variant has been identified actually very early on. The difficulty to understand how this could cause disease is if you imagine that 50% of the people harbor this gene variant, and how such a common variant can lead to disease susceptibility hasn't been clear for quite a while. And it has been recently found that this variant is actually providing a cleavage site for the gene, and then people who smoke have increased cellular stress, and in certain cell types, for instance, in Paneth cells that produce antimicrobial peptides, this leads to a deletion of this gene product, and this suggests how an environmental factor can have a significant impact on a gene, and how this can contribute to disease susceptibility. In addition to our understanding how genes can cause disease, we have increasingly an understanding why disease variants occur in the population. And this is still a bit of a conundrum in many different fields. For instance, you might ask yourself why there is such a high frequency of cystic fibrosis in the population. 1 in 20 people in England have the gene variant that can predispose to cystic fibrosis. Similarly, we do have some gene variants that are more common than expected that can predispose to inflammatory bowel disease. Just because these variants can cause, if untreated, even in IBD, an extreme severe disease, it is not clear why those variants can accumulate in the population. And so we have, for instance, investigated the frequency, how IL-10 receptor variants are present in different parts of the world. And a postdoc in the lab, Dominic Aschenbrenner, has seen that these variants in particular accumulate in East Asia, in areas in China, in Japan, in Korea in particular. And this would not be explained, for instance, by sort of the just sort of a variant that is present in a certain region, in certain villages, a very secluded sort of population, because many different variants, over 15 genetic variants, are present in this region. That suggests that there must be environmental pressures that have led to accumulation of these variants. And one hypothesis that we have is, for instance, that certain pathogens use an accumulation of IL-10 as an regulatory cytokines to survive in the host, and they can have detrimental impact in people. And so it might have been, to a certain age, of an advantage to have at least one copy of a less functioning IL-10 receptor. That led to a heterozygous advantage, to an accumulation of this variant, and that sort of had been a protective effect in the heterozygotes, even if in homozygosity this can have a severe effect and children die from this condition. So what I wanted to do is to say, well, genes never act on their own. They always act in an environmental context. Some context sort of is driven by evolution, and some is driven by the changed lifestyle that our westernized our living conditions are. And our genetics is not changing, but it has evolved over time. It has accumulated. It has not necessarily evolved for the conditions we currently live in, for instance, processed food, use of antibiotics, changing microbiome as well. And also, this has been largely beneficial in the last 100 years for our overall survival as a population. It harbors some risk factors to develop immune-mediated diseases. With this said, I'd like to thank the group collaborators here in Oxford and worldwide. And I hand over back to Chris.
Thanks so much, Tom. So you can see the beautiful interaction, the usual thing, host versus environment, and genes and environment, nature and nurture. Callie, you're going to take over now and tell us, actually continuing the theme of inflammatory bowel disease, because that's actually quite a nice one where this genes environment interaction play out. But if you ever wanted to know anything about the cellular basis of disease, listen to this story. It's pretty cool.
Thank you, Chris. Can everyone see the slides okay? Yes, fantastic. Great. Thank you. So I think given technological advances, we're now very uniquely positioned to be able to understand human anatomy, pathophysiology, inflammation at really an unprecedented depth. I like to think of us sort of studying inflammatory diseases in kind of multiple dimensions, if you will. So we're looking really across different diseases, across space, so whether that's within tissues, between different tissues, and over time as well. So thinking a little bit about how chronic inflammation changes as disease progresses, but also what happens upon perturbation, so before and after drug administration, for instance. Through these kinds of cross comparisons, essentially we're going from genes to cells and tissues through to tissue function. And these kinds of cross comparisons allow us hopefully to be able to gain some insights in terms of shared etiology across diseases. That could be shared genetic etiology or otherwise. We can begin to look into cross disease pathology upon perturbation, for instance. And ultimately, we can begin to integrate all this information, so integrating the genetics, spatiotemporal profiling, insights in terms of cellular and tissue function, hopefully in terms of being able to derive some translational benefit from these kinds of analyses. And to be able to kind of illustrate how we can go really from cells and tissues through to hopefully beginning to get these insights in terms of translational benefit, I'm going to take you through one specific study, as Chris was mentioning, in the context of inflammatory bowel disease. So Holm obviously very nicely introduced IBD, and just to kind of note that, of course, when we're thinking particularly about adult inflammatory bowel disease, we have this interaction between genetic factors and environmental factors that ultimately lead to immune dysregulation and therefore chronic inflammation and a range of sort of downstream implications. And if we look at the two most common forms of adult IBD, we have Crohn's disease and ulcerative colitis, and these differ to an extent. So, for example, in Crohn's disease, when we look at the pathophysiology, this tends to be sort of patchier, affecting both the small intestine and the large intestine, whereas in ulcerative colitis, we have more of a continuum of pathophysiology along the colon or the large intestine. Nevertheless, despite the fact that we know that we have some of these differences, there are also some key commonalities. And one aspect that's in common that we've known about for a long time is the involvement of the cytokine TNF, obviously a pro-inflammatory cytokine, which can be derived from a range of different cell types that have been implicated in IBD, and can equally have a wide range of different targets and a range of sort of effector mechanisms and downstream consequences that promote pathology. Now, given that we've known about TNF for a long time, it's no surprise that anti-TNF therapy is the most commonly used first-line biologic class in IBD. So drugs targeting TNF were first approved 20 years ago or so. However, there is a bit of a challenge in that about 40% of patients don't respond to anti-TNF therapy. Now, thankfully, today there's a range of other types of biologics that patients can receive, targeting a range of other cytokines and pathways. And whilst this is obviously a very good thing, it also poses a significant challenge. Because, well, if a patient is not responding to anti-TNF, what sort of drug should they be going on to instead? And so we hypothesized that if you can understand the cellular impact of drug-dependent perturbation, then this might be very important in order to help guide stratification and drug sequencing. So trying to kind of rationalize through a cellular basis or the cellular understanding of disease, which therapy a patient should receive and when. And so to do this, we had an excellent team of people in Oxford and collaborators in Birmingham and Celsius. This study was led along with Chris Holm and Simon Travis, and was really driven by a fantastic clinical fellow, Tom Thomas. So in terms of the design of this study, the aim was to perform longitudinal profiling of biologic-naive Crohn's disease and ulcerative colitis patients before and after therapy with adalimumab, so anti-TNF. And so we took biopsies across different regions of the gut before treatment and also after treatment in these patients. The patients could be stratified into individuals who went into remission after treatment and those who did not. And taking this full set of samples, which amounted to just over 200 samples in total, we performed high resolutions of single cell profiling of the gut biopsies, along with histopathology and imaging and spatial transcriptomics. And so ultimately, we generated a very large therapeutic atlas of IBD. So we were looking at about 1 million cells that we could classify into over 100 different cell subsets across kind of 8 main cellular compartments that we have in the gut. So we could identify specific subpopulations of immune cells, stromal cells, vascular cells, epithelial cells. And the exact details of precisely individual cell types is not important here in terms of this slide, but really I think the take-home message here is that we have really a high level of cellular granularity that allows for fundamental questions about disease pathophysiology and the impact of treatment to be addressed in depth. And so what were some of the questions that we wanted to try and address with these kinds of data? We wanted firstly to understand how the inflammatory landscape of Crohn's and UC compares. We wanted to know what cell types and pathways really characterize non-remission in IBD after anti-TNF. And we also wanted to understand whether pre-treatment cellular signatures were associated with remission after therapy. So in terms of this first question, we began to explore the relative stoichiometry of different cell types in Crohn's and ulcerative colitis. And whilst we saw many commonalities in terms of what characterizes kind of an inflamed Crohn's disease or ulcerative colitis biopsy, we also did note some specific differences. And so, for example, we found that in inflamed samples from Crohn's patients, we have a specific enrichment of, for example, Th1 cells. So these are CD4 cells that are producing interferon gamma, as well as a certain population of enterocytes, whereas conversely in ulcerative colitis, we found an increase in slightly different types of immune cells, so certain CD8 subpopulations, the Th17 cells that are producing IL-17, and also a population of plasma cells that are producing IgG antibodies. And having seen some of these differences with respect to kind of the stoichiometry of particular cell types between the two diseases, we then wanted to understand how this might relate to the spatial localization within the tissue. And so to do this, we performed spatial transcriptomics, and this work was also validated at the protein level with imaging analyses. And through our spatial transcriptomics approaches, we found that there are specific inflammatory states and gene expression signatures that map really to distinct pathophysiological niches within the gut. So for example, in terms of these types of niches, we found granulomas specifically in Crohn's disease. And then in both diseases, we found lymphoid aggregates, so where we see sort of an accumulation of T, B cells, plasma cells, as well as other areas of tissue damage in both diseases that were characterized, for example, by changes in terms of gene expression profiles within stromal cells, presence of neutrophils, and so on. So now, having characterized the inflammatory landscape of both diseases, obviously what we were really keen to look at next was the fact that we had sort of longitudinal samples, and to really understand what was happening before and after perturbation, i.e. anti-TNF therapy, in our different groups of individuals, particularly looking at non-remission and remission patients. So if we look firstly at the non-remission patients, what was particularly striking here were the changes that we saw when we were looking at this longitudinal comparison of samples before treatment versus after treatment. And what we really found was that after treatment, we're seeing a really substantial worsening of disease despite anti-TNF therapy, which really does suggest that if you don't get the treatment right initially, this does have substantial consequences for patients. Inflammation is not static, it is getting worse. And so ultimately what we found is that we had niche-specific changes really in the multicellular inflammatory processes that are underpinning non-remission after anti-TNF. For instance, in Crohn's disease, we saw changes particularly in the myeloid compartment, for example. In ulcerative colitis, we saw changes across multiple different cell types. But in both cases, we did see changes in terms of, for example, interferon signaling. So we saw increases in interferon signaling post-treatment in these non-remission patients across different cell types. And for instance, what we might be able to derive from these sort of patterns of change is that potentially maybe coming in with JAK inhibition as a therapeutic option for non-remission might be the way to go for these sorts of patients. Now, having looked at the non-remission patients, the next thing that we also wanted to do is to see what was happening in those patients who actually did go into remission. And so here, if we think about what's happening after treatment in these patients, as we might expect, essentially inflammation is going down. But what was rather more interesting is what's happening before treatment. So when we looked at the pre-treatment individuals who would go on to have remission after anti-TNF, we found that we could identify very specific pro-remission signatures, we think, which are shown here, for example, in blue. And so what we found, for instance, was that goblet cells were specifically expanded pre-treatment in the remission group, well above the sort of healthy state. And not only were these cells increased in abundance, they were also, their phenotype was also altered. So we could see that we had an increased sort of functional capacity in these cells because we could find evidence, for instance, of higher mucin gene expression. And so what this is suggesting is that epithelial integrity may actually be particularly key in terms of promoting drug-dependent remission. And so just to kind of summarize, going from this sort of large-scale atlasing effort, leveraging technologies that allow us to profile cells at a very high resolution, we arrive at this scenario where we could say that in terms of IBD remission, just dampening down inflammation isn't enough. So in order to achieve IBD remission, you need to not only be able to dampen inflammation, but this needs to be happening in a context where you have pro-resolving pathways being upregulated and occurring. And it's really that balance that allows you to achieve IBD remission after treatment. So where are we going with this sort of work in terms of being able to exploit its full, hopefully translational potential? So we're now in the process of following up the pre-treatment pro-remission signatures for a biomarker discovery program, for instance. And we're now also assessing the impact of JAK inhibition and as well as other therapeutic approaches in IBD by now generating cross-treatment atlases in Crohn's disease and also in ulcerative colitis to begin to really understand, does remission look similar and do sort of pro-remission signatures look the same across different types of therapeutics? And do we see this evidence that actually individuals who have not gone into remission after anti-TNF, are they actually benefiting from JAK inhibition, for example? And as I mentioned initially at the start of my talk, we're actually looking across tissues, across diseases. So we're now extending this approach beyond inflammatory bowel disease, looking at other immune-mediated diseases and looking at parallels across these diseases. So for instance, we've begun to do this now with rheumatoid arthritis as well, where we can see some interesting patterns in terms of, again, this interferon response signatures kind of coming up, and interesting patterns, again, in terms of drug response and non-response. And so finally, I'd like to thank our many other collaborators in Oxford and beyond, our funders, and of course, the patients without whom this work wouldn't be possible. Thank you.
Fantastic. So you can see how it nicely leads from the gene and environment to the cell. And so what I'm going to do now is just answer the so what question, which hopefully we'll bring up now here. So I'm going to assume that on the call, we have about 35 people, I think, joined, that many of you are physicians. Not all of you will be, of course, but if you're a physician, you would know the answer to this question. What have these four organs got in common? So we've got here a joint, the hands, an eye, a gut, and the skin. And we know that there are diseases that are comorbid with these. So if you're a gastroenterologist, you say, the extra intestinal manifestations of ulcerative colitis and Crohn's are an arthritis. And if you're a rheumatologist, you'd say, well, the extra articular manifestations are of uveitis or psoriatic arthritis. But they all share commonality. There's something about these diseases that links them. And in the same way, ulcerative colitis is linked to certain liver diseases. And we've always assumed that that link must be something to do with infection or shared, infectious agents. But it could be that actually what they've got in common is a common cell type that's responding in a certain way to damage. And so I want to, in addition to servicing the clinicians, the physicians, what about when it comes to drug discovery and when it comes to the way we practice medicine? Because if we're going to look at inflammation across tissues, we have to be able to understand what's common about that inflammation and what's unique. So that, for example, we know that there are a certain number of genes, 20,000, that make up a human being. And when we make a drug, we target something that's been made by a gene. Sometimes we target the gene itself. But the gene is making something on a cell that we can then target or is secreted by a cell or in some way affects any of these processes that we see here. On this side are all the specialties, if you like, the organs that we have diseases in. So people will say, well, the indication that we want to use this drug for is arthritis and specifically rheumatoid arthritis, or inflammatory bowel disease and specifically Crohn's. And we know there are about 80 organs, but we've got a problem because when we go from genes to organs, we don't know the bit in the middle, which is how many different cell types are required to go from that gene to that organ. So if you're a hematologist, you're in luck because the gene called hemoglobin manifests in a red blood cell that is in the organ of the blood that leads to anemia. It's a beautiful straight line. But for most diseases, I guess infectious diseases are also where we can be causal, metabolic diseases, but almost all inflammatory diseases, we don't know the link between the gene, the cell, and the organ. And that's what today's webinar is trying to address. What's that cellular commonality? So that's the problem when you're trying to design a drug, because you make a brilliant drug, it targets factor X. You say factor X is implicated in disease Y, therefore X must be involved with Y. Well, why should it be? there may be other methods or mechanisms, and this is probably why we fail so often when we try to take drugs into certain indications. The problem now with clinical practice, for the clinicians on board, is that when we look at the disease indications here, this is rheumatoid, Sjögren's, salivary glands, spondyloarthropathy, inflammatory bowel disease, we're very descriptive in the way we describe them. We're rather like Darwin describing what he saw on the Galapagos without knowing that the cause behind all of this is actually DNA. And the rules of understanding how DNA are put together, of course, transformed medicine 60 years ago. But the problem we have in addition is that the readouts we use for clinical efficacy are so imprecise compared to the precision that goes into making the drug that we use things like disease activity scores, patient global assessment, X-ray damage for rheumatoid. And in essence, each of these things have three components: a activity index, a damage index, and a patient or physician global assessment. But the irony is that underpinning all these diseases are common cell types. Who would have thought that depleting B cells would not only help rheumatoid, but also help diabetes and in some cases MS. And of course, that there's been a massive interest now in CAR T-cell therapy to treat cancers as well as some of these diseases we've got here. So there are two problems. The problem is that we don't understand the marker and mechanism and how they're linked, and we don't know the cellular basis of our diseases. So how do we solve this problem? And what we've tried to do in the consortium who are talking to you today is to test the hypothesis that there may be a common cellular periodic table. So I just need to show you this picture, and you immediately know that it's actually of the periodic table, and you know that the rules by which one element goes with another element to make a compound are well known. Imagine we didn't know the periodic table. We didn't know that water was H2O. How could we function in the lived world? And now you're asking physicians to function without knowing the cellular basis of their diseases. And that's where the problem lies. So the hope is that the Human Cell Atlas with the Human Genome Project will lead to a better understanding of how diseases evolve through their cellular components. So here's the concept that in addition to having chemical elements, you have cellular elements that have rules by how they interact. So certain rules like how immune cells interact, how muscle cells interact with nerves, and could we imagine that they're arranged in families in a similar way to the way the chemical elements, and therefore it won't be random which cell goes with which other cell. There'll be rules about how that happens, just like a certain nerve goes with a certain muscle. And if we understand that at a tissue level, just as Callie showed you, we would understand now how the drugs, which might act in the similar analogy here to catalysts, would break down certain combinations of cells and allow us to understand how we can get tissue back to normal. So I want to give you real examples of how knowledge of that cellular clinical periodic table has been used in the design of trials that are breaking down the way we commonly think about how we do them. So in a traditional trial, you have one drug, one disease, one outcome. Suppose you have a drug, but you're not quite sure which disease indication it might work in, but you know that that drug targets a particular cell. So here's an example where this drug that we did in collaboration with J&J targeted a molecule called PD-1. Now you'll know about that because that's the one that's involved in checkpoint inhibition in cancer. But the idea was if you could agonize this, you might actually improve the opposite of cancer, which was inflammatory diseases. But in order to do the study, we needed to choose diseases because the idea was, well, which of these diseases might it work in, where this cell type was present. And here it is, this white-looking cell type, these T peripheral helper cells that express PD-1. And this trial was a collaboration across 4 centers in the UK. And it's, again, similar to what Callie showed you. You take a biopsy, you give the drug, and you take a biopsy again, and you ask, has that drug done anything to that tissue? And in this case, the readout was depletion of these, what we think now are pathogenic TPH cells. The equivalent cell in the lymph node is called a TFH, or follicular helper cell. And you can see that only in rheumatoid do you deplete these cells, but you don't in Sjögren's. So this is really helpful because what it is saying, look, the rule by which rheumatoid is happening, we can affect these cells, we can kill them off in the rheumatoid synovium, but we can't do it in the salivary gland. And if you start to think, well, what's different about the salivary gland to synovium? Of course, the salivary gland has microbiome, it has an epithelial structure that the synovium doesn't. So here's an understanding of how that clinical periodic table might be helpful and how we're going to move forward into the future. Well, can you imagine a future where we could look on your iPhone and say, rather like you would ask AI or Google, can you tell me who makes and who responds to a particular drug in a particular tissue? So Callie and her and Steve Taylor and a number of others, as part of these collaborations that we've been doing with our pharma colleagues, is able now to look at these atlases, many millions of cells, pretty much as high, getting close to 10% of what actually is in the Chan Zuckerberg initiative across a range of donors, and ask, what is the data that we have on them, clinical metadata, as well as this single cell? Can we integrate that data? Easier said than done. Will it now allow what we call this multidimensional view to see this data in real time, to see which cell types are going with which cell types, and understand and predict what a drug might do to break down or affect those cells in some way? And here's the kind of the proof of the pudding that actually all of this might lead to something. You, as physicians, and even if you aren't, you will know that defining normal is really important. What is health, right? So if I have somebody who's 18 or 80 or 18, how I'm going to define what their health looks like is different, because of course there's age and so on. But what we've been able to do is to define it at a cellular level. So up here in these different colors are all these, this is Callie's brilliant work, joint diseases. So you can see health is in blue, rheumatoid is in red, psoriatic arthritis is in green, and so on and so forth here. And then each of these columns is a patient, and within each of these columns are the cell types that make up that cellular component. So view it like the stoichiometry. This could be sodium bicarbonate. So it's got, this patient has rheumatoid is red. Here's the amount of fibroblasts, here's the amount of endothelial cells, here's the number of T cells, and so on. And if you look at that for the disease we call rheumatoid, can you see that actually it's not one disease? There's this type of rheumatoid, and probably this type of rheumatoid. So there's at least two main types, ones where there are a lot of immune cells and ones where there aren't. But even within the immune cells, these fit into what we've now described as lymphoid-rich, myeloid-rich, and so-called fibroid or lacking lymphocytes. And they're very different to what health looks like. Health actually is lots of these fibroblasts and very few lymphocytes. But now look at another disease called psoriatic arthritis or osteoarthritis. This is one disease. It's on a spectrum. And so PSA is actually further away in terms of its disease characteristics to rheumatoid and much more like osteoarthritis. And of course, if you think about it, that's what we see. New bone formation, distal interphalangeal joints, and the route back to health may be very different. The type of drug that you might want to use here will be fundamentally different to what might be happening in rheumatoid. Let's look at the bowel now. Here is, sorry, the colors are slightly different because now we have epithelial structures, which we don't have here. Health is here. This is normal, non-inflamed, and now you can see inflammation is dominated by infiltrations of these leukocytes as well as loss of epithelial cells. But yet again, even though Crohn's and ulcerative colitis have a similar kind of look, we know they're different at a single cell level. There doesn't seem to be these big differences like in rheumatoid. It looks like it's on a spectrum again. And so this is important when we're going to try to say, well, how could we design trials where the commonality is a cell type, but now the readout of whether the drug is working is how far you get the diseased patient back to health. So we're going to call this distance to normal. And statisticians love this because it's a metric of efficacy, but you can use it across tissues, just like you would use CRP as a metric for inflammation across diseases. And the second thing that the atlases allow you to do is what Callie was showing you, guiding therapeutic sequencing. So what drives serial non-remission? And this is another clinical fellow who's doing a follow-on study to Taurus. We tend to name our studies after signs of the zodiac. But in this case now, we're taking all current standard treatments and taking a biopsy, giving the treatment. If they respond, that's great. If they don't, we switch drugs, take another biopsy, switch drugs, take another biopsy. And this allows us to say what's happening at a cellular level in people who are undergoing serial non-remission. So guiding therapeutic sequencing, allowing us to understand the cellular causality are two real tangible outcomes that inform us with the knowledge of the periodic table. So these are the guys. We're lucky in Oxford to have one of our BRC themes, inflammation across tissues, where we work on I haven't shown you data on the skin, but the gut, the joint, the skin, and of course the eyes. And just because I'm a naughty boy, I'm going to suggest that those of you who are not just clinicians, but scientists who want to do a really good DPhil, come and work with us. We're open now. Actually, the lines are open, so to speak. Go online, and we have our Kennedy Prize studentships, as well as a number of other ones. And so do consider joining this team, because I think you'll not only have fun, but actually you'll help break down these silos which don't help anybody. So I'm going to stop at that point, and we now have, brilliantly, we have time, 15 minutes for questions.
Thanks, Chris. Great. Well, look, if you have a question, can you put it in the question Q&A box, please? And we'll attempt to answer them. Or put them in the chat, but the Q&A box is open and available. So far away. Meanwhile, I might just kick off. And I just wonder, I mean, as a general question, obviously you can do these marvelous single cell studies and pick up signatures, but that's not going to be that easy to apply to patient prognosis and so forth. So which of the steps, or what are the steps that you could use to kind of simplify it, to extract that information to, say, split your patient according to what drug they're going to need? Have you got a sort of pathway for doing
that's a good point. And I'm going to ask Callie to have a go at answering that. Hidden in that question is another part, which is how good does the cellular classifier match onto the clinical classifier that we already use? Because ultimately, we can't biopsy everybody, right? So we need to understand. The null hypothesis is it won't. That's a bit of a bummer, because we were used to using the clinical classifiers. But think about it, in cancer, we don't use clinical classifiers. So I'm going to be controversial and say we don't, in cancer, treat the patient in front of us, we treat the disease, because then we can cure the patient in front of us. So patient-reported outcome measures, they're brilliant and so on, and we want to use them. But actually, I'm not sure, and I know I'm being controversial deliberately, whether the clinical classifiers will ever match to the cellular. And then we have to understand which is truth. Is the cellular classifier better than the clinical one? Take over, Callie, and rescue me from my naughtiness.
Yes. So I would say that I think as we're getting, generating more data, I think certainly the hope would be that we don't sort of have to indefinitely be doing single cell and then sort of spatial omics approaches. That would be clearly impractical, from in terms of actually being able to apply some of that knowledge in real time in many cases. And so I think the idea really is to try and see, can we, from these kinds of analyses, obtain more minimal signatures that will be of fundamental value in terms of prognosis. So I think I would say that we're not there yet, I think, in many instances, but that's certainly what we're heading towards as we're kind of generating these larger data sets, and also beginning to apply, for example, in leveraging machine learning AI approaches to better understand, can we build up a predictive framework, and alongside that, how do we do this in practice? So for example, taking the example actually of the changes in, say, goblet cell function that we saw in the IBD patients who went on to have remission after anti-TNF. That's an interesting signature, but you could argue that it's a little bit in impractical one to try and measure in patients. You'd have to be taking a biopsy and then doing some sort of omics, whether that's, even if it's a bulk sequencing or qPCR or what have you, to be able to try and identify individuals that have these kinds of pro-remission signatures. And so one of the things that we're working on is trying to see, are there alternative approaches? So for example, leveraging the knowledge of the kind of signatures that we want to be looking at, and using other approaches like fecal washes rather than actually taking biopsies, for example, to begin to get a sense of, is there a possibility to be able to kind of refine our approaches so that we don't have to be doing single cell all the time, and can actually be using more practical methods that can be implemented in the clinic.
But I guess there might be, from the thousands of gene expression markers that you can get per cell, Do you think there might be still a molecular, even if it's slightly complicated, but molecular approaches are still, quite clinically applicable? So do you think you could just distill some of these things down to a small panel or a single gene? How's that going?
Yes, exactly. So, and that's exactly what we're trying to do. So we're trying to kind of improve in terms of the sampling method that we can go for. So do we need to always take biopsies, or can we go for an alternative approach? But very much, even if, for example, we can manage to do this, say, with fecal washes rather than biopsies, this is still going to require some analysis of a molecular signature. But again, rather than going for maybe a full transcriptomic signature, we want to refine it down to a specific subset of genes that are perhaps the most robust in terms of capturing these kinds of pro-emission signatures, for instance. Yeah.
Can I ask this one question of Holm, actually? Holm, one of the interesting things I kind of put up was that maybe Crohn's and ulcerative colitis could be on a spectrum, but I'm not sure if we had bigger numbers of patients in the single cell data, whether that would hold. Because what you show is that the different genetic environmental risks, maybe there's more than one type of ulcerative colitis or one type of Crohn's. Can you tell us a little bit about the overlap between those two kind of diseases in terms of genes, environment? And also, I'm kind of priming you here, what the heck is a plasma cell doing in the colon?
So, yeah, I mean, this brings in principle sort of this fundamental sort of question what we call a disease. And so these two diseases, Crohn's disease, I mean, dating back sort of nearly 100 years since the initial description by Beryl Crohn and described sort of in earlier case reports earlier, ulcerative colitis at least so 100 years ago, and all dating on H&E histology, so with two different stains, and the classical sort of description of a transmural discontinued sort of disease that can come with granuloma or with fistulizing sort of disease, structuring disease in Crohn's disease, and a superficial lamina propria infiltration that is continuous, that is just restricted to the colon in ulcerative colitis. Now, what we have seen and genetic disorders tell us that there are many different reasons or many different sort of genetic causes that can lead to an end result that looks like Crohn's disease with fistulizing sort of disease or ulcerative colitis. And what we increasingly now understand is that there are certain modules, for instance, defective IL-10 signaling, or defective neutrophil function, or defective macrophage metabolism, that all can lead to a features that we call Crohn's disease, and a pathologist would classify as Crohn's disease, and some epithelial defects or some immune regulation sort of defects that are more similar to ulcerative colitis. So overall, it looks like what we call Crohn's disease and ulcerative colitis are very much of a spectrum. There could be extreme cases, for instance, and as one of the exemplar, we have, for instance, shown that even autoantibodies, for instance, against the immunoregulatory cytokine IL-10, that is, can block and neutralize IL-10 activity. So it's not always a genetic condition. That's what we call a phenocopy, an autoimmune phenocopy, and that can lead to a Crohn's disease like disease over time. And we described it in a patient with infantile onset, and as one exemplar, we could show that targeting B cells, for instance, by blocking CD20 and depleting short-lived plasma cells, is a proof of concept that in principle, sort of even B cells sort of can be pathogenic in Crohn's disease, not only in Sjögren's syndrome or in some forms of rheumatoid arthritis, and that there are some forms of IBDs that are probably more similar to the rheumatological diseases. And that provides this new concept that there are probably a limited number of key mechanisms that can be triggered by either genetic or autoimmune or environmental sort of factors that then downstream lead to certain signatures. And these where maybe the single cell signatures come into play, where we have sort of subgroups of response modes that might be either targeted by their cellular basis or by the mechanisms that lead to cellular dysfunction. And in the end, it's all a clinical trial that needs to prove whether this is correct or not. It's the intervention. But if we have shown this for a key subset, then it's likely that we can use this as better biomarkers than a classical CRP or a fecal calprotectin or a thrombocyte number that we currently use to substratify patients.
Thanks, Holm. Paul, I don't know if you want any more.
I might just ask one more. We got just a couple more minutes. I can't see any more questions, but I'm curious about the finding that things got worse. I think it's Callie's finding, but perhaps you can all comment on it. So if you don't the patient doesn't have a good benefit from the anti-TNF, there were some features that the disease wasn't static, but the implication was you were sort of accelerating things. And obviously the sort of, like, for a bunch of reasons, you want to treat people with the right drug first, but there is an implication that if you get it wrong, you've got a downstream worsening of disease. And I don't know, is that essentially coming to be seen as true? And if so, is it in IBD, and beyond IBD, is it also seen in the rheumatological world? So I guess it's a question for all three of you.
So, Paul, the problem is one of time. Where are you in the temporal sequence of this patient's journey? And that's the difficult part. And we use sometimes experiments of nature. So people being treated for checkpoint inhibition, getting inflammatory diseases, or people coming off their drug. But I guess, Callie, you've got kind of a lot of experience of looking at that temporal component. And we don't want to make things worse, of course. But it's certain drugs may certainly not. It's not that they're making it worse, but they're certainly not making it better.
Well, I wondered whether there's an implication it might make it worse, because
that's the concern.
Counter-regulation going on.
Yeah, exactly. So, Callie, you exactly the point, Paul.
So I would say that from the data so far, I mean, in some ways, the perfect comparator, I suppose, would be an untreated patient over time, which obviously we wouldn't be able to look at to know whether we're seeing really an accelerating effect. But I think once we've done, for example, the Sagittarius study that Chris was showing, where we're comparing across different types of drugs, I think we'll have a bit of a better sense of whether in certain instances, do we almost have an acceleration, or are we just capturing kind of features of time? And actually, if you're not managing to dampen down certain pathways, inevitably things are getting worse. So I don't know if we have enough evidence right now to say that we actually, in some instances, might have an acceleration. I think once we've got some additional data, we may be able to begin to kind of try and address that question. But I think either way, as Chris was saying, I mean, I think if we don't get it right, even if we're not making things actively worse, we're certainly not making them better, and that it will likely have consequences.
Hard to tell. There's one final question, I think, from Linda in the chat. And it says, have you tried applying, I think this is for you, Callie, probably, but it could be anybody. Have you tried applying machine learning methods to integrate different data sets, multiomics, EHR, etc.? What's the challenge when you try and do this?
So the answer is yes, we are starting to do this indeed. I would say that part of this, so there are multiple challenges. Part of this is one of data and the scale of data, and in some ways, if you will, how good your data is. So from a multi-omic sort of single cell perspective, I would say that the data sets are now getting to a point where they're getting large enough to be able to apply some of these methods and to hopefully derive some insights from them. So that's something that we're actively working on right now. I think this space has moved more quickly within the context of electronic sort of health records, partly again because of the scale of the information. But I think again, it's about how when you apply these kinds of methods, what question are you really trying to address? And I think sometimes you might have a slightly different question when you're applying these methods to electronic health records versus when you're applying it to sort of cellular data. But I think as data sets grow and as some of these tools also mature more, I think we will be able to gain insights, and certainly also from the integration across different modalities. I think kind of a very active area of research right now is how to make some of these methods more explainable. It's interesting to apply them. You get certain answers, if you will, but trying to understand actually what are the key drivers that give you specific patterns in the data when you apply machine learning or AI tools, I think is very much a key question as we're now moving forward, which hopefully we'll be able to get some, I think, improved tools as we move forward in the next few years. Thank you.
Oh, thanks, Callie. And that's a great place to stop, by integrating data sets across diseases, across tissues. I think that was a sort of kind of core kernel of the what we're trying to look at. Chris, have you got any last comments? If not, we can draw to a close.
Yeah, all fine. And if any of you online or watching this later are interested, please do contact us and help us to deliver the new periodic table of cells, the physician's periodic table. That would be so cool.
Well, thank you very much. Thanks for coming. There are plenty more of these from the Association of Physicians. Any requests, also please contact us. We could organise one. So thank you very much, and have a good evening.